Good day, everybody. My name is Alejandro. I'm going to share with you the results of my undergraduate work, and the title is Frequency and Spectrum of Mutations in 7 Genes in Women with Breast Cancer from 3 cities of Colombia. This is the whole team behind this project. I work with the Infection and Cancer Group at University of Antioquia, and the director of the group is the Professor Gloria Sanchez. Breast cancer is a complex disease, which means that environmental and genetic factors contribute for the development of this type of cancer. However, around 5 to 10% of breast cancer cases are hereditary, and this is due to mutations in genes like BRCA1 and BRCA2, but there are also other genes that are involved in cell cycle and DNA repair, such as CHEK2, ATM, PLB2, and so on. The frequency of mutations in breast cancer cases depends on several factors, such as the presence of other mutations that are variants that arise from a common ancestor, the number of genes evaluated, because if you evaluate more genes, you have more chances to find this kind of mutations. But also the technique for detecting the mutations is important, because technologies like next generation sequencing really improve the sensibility of the genetic tests and the selection criteria of cases, because if you choose high risk breast cancer cases, such as those with family history of breast cancer or an earlier age of onset of the disease, you have a higher probability to find this kind of variants. Here in Colombia, only a few studies have evaluated this frequency in unselected cases, with results that vary from 1.2% to 4.2%, but these studies have mainly focused on the BRCA genes. Sometimes they have used a panel of mutations and even high-risk cases instead of unselected cases. So the aim of this study is to determine the frequency and spectrum of mutations in 7 genes in unselected breast cancer cases. For the mutation detection, we extract DNA from white blood cells, and then we sequence the exons and splicing regions of 7 genes with the Ion Torrent platform. We have 135 cases. Then we perform a quality control of raw data. Then we align the reads to a reference genome with TMAP. The Torrent variant caller was used for the variant calling, and the Ion Reporter software for the variant annotation. For the clinical classification of the variants, we use the following databases: ClinVar, 1000 Genomes, LOBD, the UMD, and we use the bioinformatic tool WInterVar that basically applies the American College of Medical Genetics and Genomics guideline to classify this. As a result, we found a frequency of carriers of 4.4%. We found in total 6 pathogenic mutations. 3 of them were intronic, 2 of them nonsense, and 1 missense. 4 of them were not previously reported here in Colombia. And we found 17 variants of uncertain significance. And 1 of them in the CHEK2 gene has a great pathogenicity according to the in silico predictors. So it's really interesting. As conclusions, we found a frequency of carriers of 4.4%. This is the first study in Colombia that evaluated other genes than BRCA in unselected breast cancer cases. We found 3 splicing variants, so it is necessary to include intron-exon boundaries in the target of the sequence, and we found that this variant in CHEK2 has a great pathogenicity in silico, so it can be reclassified, but we need to do in vitro studies to confirm the effect of this mutation at the protein level. I want to thank the Infection and Cancer Group, the University of Antioquia, and other institutions that helped us with the recruitment and analysis of the data. Of course, the Ministry of Science and Technology for funding this project, and especially to my professor and director of the Infection and Cancer Group, the Professor Gloria Sanchez.